All right, I'd like to go ahead and get started with the immunogenicity education session. Just a few points to remind everyone. Please silence your cell phones and mobile devices so that you don't interrupt the speaker or hinder any of the audience during the talks. Also, again, photography and filming are prohibited for both here, but also for throughout the conference. We no longer provide paper evaluations, but there is an opportunity to provide an evaluation, so attendees can evaluate each session through the conference mobile app, and if you haven't downloaded that, one can do that in the usual ways. So just to give you a little bit of a sense for what the immunogenicity session is going to cover, with the increasing clinical translation of gene and cell therapies, but particularly gene therapies, one of the issues that has arisen is I would say, an increasing encounters with immune responses against both the vector capsids and also against transgenes that are being expressed by those vectors. And so in today's session, you're going to hear from speakers that are covering complementary aspects of how people are thinking about those. So Dr. Herzog is going to cover a little bit on aspects of AAV vector biology that impact on immune responses. Dr. Cabral is going to talk about some of the approaches to modifying expression from AAV vectors that can be used to also modulate immune responses. And our final speaker, Dr. Miao, will talk about a more direct way of addressing transgene immunogenicity using regulatory T cells. And with that, I'd like to start with our first speaker, Dr. Roland Herzog from both University of Indiana and also University of Florida. Roland?
Thank you very much. So, all right, my lab's new home as of 2 weeks ago. Let's see. Okay.
Yeah, they'll start that up.
Okay.
So press it's backwards. The green one.
Oh, I see. Okay. Well. Is it working? All right, there we go. So as you are aware, a lot of us are very fond of adeno-associated viral vectors because they are efficient in transferring genes into cells in vivo so that you can administer the vector directly into the experimental animal or into a patient and transfer genes into various types of tissues. And there are obviously a number of serotypes available that have different tropisms, so some are very effective for certain tissues and cell types, and you have choices. The AAV vector is a DNA virus that belongs to the parvovirus family. It's a small DNA genome that is packaged into a protein capsid, and then in the viral vector we eliminate the viral gene, so basically it just carries our therapeutic gene. And so AAV vectors have enjoyed quite a bit of success in very recently, including complete corrections to nearly complete to complete correction of hemophilia A and B using hepatic gene transfer. You are aware of a cure for blindness with retinal injections or subretinal injections, and also successful treatment of SMA in infant children. And so, but at the same time, we need to be aware that every time when you perform gene transfer in vivo, and in particular if you're using a virus, that the immune system will recognize this as essentially as an invading pathogen because the immune system has evolved to recognize pathogens. The way we could think about AAV being recognized as a foreign entity that's invading the body is that it carries along various molecular structures, including its genome, its capsid that's made of protein, and then subsequently, after gene transfer, if there's successful transgene expression, then there is a therapeutic protein that's being expressed. And so you could have innate immune responses that recognize the capsid or the genome, antibody and T-cell responses against the capsid, and potentially antibody and T-cell responses against your therapeutic gene product. Turns out that most of us have been, if not all of us, have been exposed to AAV one way or the other through natural infection. And so there is a prevalence of neutralizing antibodies, a pre-existing immunity in the human population. There are some differences from one serotype to another. There are also some geographical differences. And it also turns out that there is quite a bit of cross-reactivity of the antibody between different serotypes. So one person may not just be immune to one AAV capsid, but to actually to a number of AAV capsids. So that obviously is a first hurdle for gene therapy, as you have to pre-screen the patients, and somebody who has pre-existing immunity against your therapeutic vector, you may just not be able to achieve gene transfer depending on your route of administration and so forth, and the antibody titer. It is typically considered that neutralizing antibodies of a titer of above 1 to 5, you will unlikely be successful, for example, with hepatic gene transfer. These antibodies, the children are born tend to be born with antibodies that may have been resulting from maternal transfer, but then during the first year of life tend to not have antibodies against AAV, but then subsequently during childhood, these antibodies emerge. There are different ways you could try to get around this problem. One is obviously you're trying to identify capsids that are less likely neutralized or not neutralized by that particular patient if you have choices. There are also a strategy that Federico Mingosi had developed and Kathy High, which is decoy capsids, so that you actually spike your vector with empty capsids and they soak up the antibodies, get them out of the way, and then allow your therapeutic vector with its therapeutic payload to actually be able to transduce the target cell. And depending on the pre-existing titer, the amount of decoy capsids has to be adjusted. Now, obviously, once you achieved gene transfer, subsequently the body will develop a neutralizing antibody against the vector that you injected. In this case, you can't re-administer the vector, but there has been success in non-human primate studies swapping out capsids. So in hepatic gene transfer, which is one of the tougher targets because you're going in through a blood vessel, it's been shown, for example, that you can give an AAV8 vector first and later on an AAV5 vector and achieve gene transfer with AAV5. And it's also another study showed that you can go in with AAV5 first and come back later with AAV1 and thereby achieve repeat gene transfer to the liver. Another strategy that's being developed by Barry Burns' lab is ahead of time, before you give the first vector, to actually deplete the B cells using a monoclonal antibody that is B cell depleting, combining that with a drug that also attacks T cells. And under those conditions, the immune system is not able to respond to the initial vector, and you can actually potentially re-administer the same serotype using this strategy. Now, you may be aware that there has been successful re-administration of AAV vectors by subretinal injections in ocular gene transfer in patients, taking advantage, and there was no immune suppression involved, and you can use the same vector for re-administration, taking advantage of an immune privilege that exists in the anterior chamber of the eye. However, I also need to warn you that it is not that the eye cannot mount immune responses at all. There are a lot of variables, your vector dose, how you inject it, the serotype, and so forth, and there can be both innate and adaptive immune responses in the eye as well. Now, if there's no pre-existing immunity in terms of antibodies, you administer your vector and it gets into your tissue. The very first rapid response that the immune system has is the innate response And it's been shown for AAV that the capsid can be recognized by a toll-like receptor called a TLR2, and then the AAV genome is recognized by TLR9, which is an endosomal DNA receptor. It's been shown that the signaling through TLR9 will be influenced by the genome configuration, whether it's a single-stranded AAV or a self-complementary, and also by the CpG contents of the viral genome. And then the question arises from what results of this early innate immune recognition and which kind of downstream responses may be impacted by it. Now, the innate immune response to AAV has probably been best studied in the liver. That goes back to Dan Maruvi's work from years ago, and we followed up on that. The response is very rapid. Within 1 to 2 hours, you get infiltrates in the tissue, neutrophils, macrophages, and K cells. But it's also a very weak response that subsides very quickly. And therefore, for a while, it's been assumed that innate immune responses to AAV are just so weak that we don't need to worry about them. But I'll show you later that they do have consequences. One of them is there's actually somewhat, if you go into a TLR, so this inflammation in the liver seems to be dependent on TLR9 sensing of the AAV genome. So in TLR9 deficient mice, you don't see that, or if you block TLR9 with an oligonucleotide that is inhibitory to the receptor, you can block this response. And when you do that, you actually get somewhat elevated levels of transgene expression. I think we heard some more exciting applications of that this morning. So that's, in a nutshell, the innate immune response and what we know about it at this point. In terms of subsequent antigen-specific responses to the AAV vector or its transgene, well, AAV actually became popular in the mid-90s as a vector for in vivo gene transfer precisely because, in contrast to the prevalent adenoviral vector that was used at the time, that AAV was such a poor activator of CD8 plus T cells. So you go in with an adenoviral vector into a mouse muscle shown here expressing beta-galactosidase. Very quickly the expression is wiped out because CD8 cells, cytotoxic T lymphocytes, migrate into the tissue and kill all the muscle fibers expressing the LAG-Z. You can do the same thing with AAV and you get expression of LAG-Z for the lifetime of the mouse without the CD8 cell infiltrates. So we thought all problems solved as transcellular immune responses. And then it turns out that in clinical trials, CD8 T cell responses have been observed against AAV-encoded transgene products such as dystrophin, microdystrophin, which was very interesting. That were both responses, they seemed to be de novo responses in patients that had certain mutations so that they were not tolerant to the sequences of the therapeutic microdystrophin, but there were also seemed to be pre-existing responses that are an underlying problem of the disease. And another interesting result was that even in patients, in this case alpha-1 antitrypsin deficiency, that had missense mutations, there was an unexpected CD8 cell response against alpha-1 in 1 or 2 patients. And when the investigators looked more closely, they were surprised that the T cell response was against an epitope that was not where the therapeutic gene and the mutation differed, but it was actually against the polymorphic site, which is interesting. So that is something you need to keep in mind, that humans are polymorphic in their sequences, and your therapeutic gene is going to have the same sequence for all patients. Another big surprise was that there were CD8 cell responses against the AAV capsid that have been observed, work from, again, Kathy Hine and Federico Mingosi. And in hepatic gene transfer of an AAV2 factor 9 vector that was initially described, it's since been shown for other capsids as well, that you can have a liver enzyme release and loss of expression. So basically hepatocytes seem to be turning over, and they're most likely being killed by CD8 cells that are specific to AAV capsid. It is not entirely clear if those are primary or memory responses, but a further analysis has shown that humans do tend to harbor CD8 cells that recognize AAV capsid, at least adults, and that there is cross-reactivity, or that some of these epitopes are conserved between different serotypes. One way to suppress this response is to use steroid drugs, a strategy initially developed by Nathwani and Davidoff and adapted by others and subsequently. Well, what you can see here is that the steroid treatment was successful in blocking the CD8, well, I need to get back and tell you that the transaminases, so the liver enzyme levels, are basically used here as a biomarker for the response against the liver. So as soon as liver enzymes were rising, you can give this steroid drug, and then and that suppresses the CD8 cell response, and you can rescue your transgene expression. But you have to do it very quickly. So you have to monitor frequently, and as soon as you see the rise in liver enzymes, you have to start giving the immune suppression, because otherwise, if you are a little bit late, you won't rescue all of the therapeutic expression, because even if you subsequently suppress the CD8 cells, whichever cells they have already destroyed, they're gone, and so there's no way to rescue those. Those CD8 T cells to capsids have also been observed in muscle gene transfer, although there, interestingly, they seem to over time resolve, and most of the expression was preserved, and the muscle tissue actually started to be more populated with regulatory T cells that may have played a role in downregulating the response. So how do you how does a cell that's transduced with AAV become a target for cytotoxic T lymphocytes? Because after all, our vectors do not express AAV capsid, but we have the input capsid, and that is most likely related to the biology of AAV, which is able to infect the cell by going in through endosomal compartment, but then it needs to escape the endosome and be rerouted through the cytoplasm in order to get to the nucleus. And during that process, some of the AAV capsid is phosphorylated, and that serves as a signal for ubiquitination, and you can, that targets those capsids for degradation by proteasome, and proteasomal degradation will lead to MHC class I presentation of capsid-derived peptides on the cell surface, and now this cell becomes basically flagged for potentially being attacked by a CD8 cell that's specific to capsid. I think work from Roland Herzog's lab has shown that if you eliminate some of these phosphorylation sites, that you can avoid some of these MHC1 presentation and also achieve better transduction because more of the virus goes to the nucleus, or you can go further downstream and you have the same effect with proteasome inhibitors. Okay, so here's our current model based on various studies that we and others have performed on how CD8 cells to AAV capsids are activated. So that goes to the question, how do the CD8 cells that then go after those transduced cells, how do they get activated in the first place? And what we believe is happening is that the AAV particle is being taken up by different types of dendritic cells that need to cooperate in order to achieve the cross-priming of the capsid-specific CD8 cell. You need the conventional dendritic cells to present the antigen to the CD8 cell, and you need signals derived from a plasmacytoid dendritic cell, which senses the AAV genome through TLR9. So you need TLR9 sensing of the AAV genome specifically in plasmacytoid dendritic cells as the signal, and then presentation by the conventional dendritic cell, which does not involve TLR9 signaling, but it presumably gets its signals through the plasmacytoid dendritic cell, and now you get activation of the CD8 cell. And so that's how the innate immune sensing of the AAV genome connects to a cellular immune response. So there are consequences of the innate immune response. On the other hand, we really do not understand yet what the signals are that lead to antibody formation. I think that's still a black box at this point. So that was response to capsid. So what is the risk of an immune response to your transgene product? It turns out there's no easy answer because there's so many variables that play in. such as the target organ, how you deliver, exactly how your protocol looks like, and how you design your vector and how much you give. And a lot of that actually impacts how much transgene expression you have. And the level of transgene expression is one critical parameter in the end for whether you get an immune response or not and how strong that response is. And then of course there are host factors such as the underlying mutation and other genetic factors, or whether an underlying disease adds additional complications such as inflammation. I think that's a very clear example here of the effect of the underlying mutation. Those were hemophilic mice that had different factor IX mutations. They were injected with an AAV factor IX vector, and those that had a gene deletion, you see the CD8 cell infiltrate, and those that have other types of mutations where there is some level of tolerance because of endogenous expression, you don't see the CD8 cell response. Work from a number of laboratories has shown, though, that when you're doing this kind of muscle gene transfer using the conventional single-stranded AAV vectors, that actually that the T cells, they infiltrate and they cause inflammation, but they're not fully functional. They tend to not ever eliminate transgene expression in your muscle. That's not true for all transgenes, but for a number. And these CD8 cells that are activated against a transgene product from a single-stranded AAV vector, they tend to express markers that are typical for exhausted T cells, so they're not fully functional, and then they apoptose in the muscle and die. So, however, if you use self-complementary vectors, then you tend to have a more functional T cell response, and now you can actually get rapid elimination of your gene expression. This kind of a response is, again, TLR9 dependent. And so work from Jim Wilson's lab has shown that if you take away these unmethylated CpG motifs that are typical for example, bacterial or viral genomes, and that are particularly good immunogenic ligand for TLR9, if you eliminate them from your expression cassette, that you can drastically reduce the risk for a CD8 cell response against your transgene product, and you could get prolonged gene expression. Now, what is the mechanism of the risk of the antibody formation against the transgene product? Again, we don't know what the innate signals are that are driving this, but we do know that that they tend to be T helper cell dependent, so you need effector CD4 cells in order to activate the B cell response. And we also know that regulatory T cells tend to be able to shut this down or suppress these responses. Again, the risk should be influenced by the underlying mutation. Here's an example of hemophilic dogs that have either a missense mutation or a null mutation in their factor IX gene. We gave them an AAV vector intramuscularly expressing K9 factor IX to match with the species. And what you see is, well, the animal with the missense mutation, you get correction of the clotting time after gene transfer, and it's sustained. The animal with the null mutation, you get correction initially and then it's lost. And the reason why that's lost is because the animal may get an antibody against factor IX. Okay, that's maybe not so surprising. But interestingly, you can do the same experiment in these null mutation dogs and get long-term expression and correction if you go into the liver instead of the muscle. So it's not only the underlying mutation, there is also the route of administration, and a work by Valder Ahuda's lab has shown that you can even use this strategy to reverse pre-existing antibody responses, in this case hemophilic dogs. And so there must be something special then about hepatic gene transfer, we believe. And hepatic gene transfer can induce immune tolerance against your therapeutic gene product. That's been shown with AAV vectors and also with lentiviral vectors. We have done much work on the mechanism for how this works. We believe much of the antigen presentation takes place in the draining lymph nodes of the liver, but also in the liver itself. You get induction of regulatory T cells that are the CD4, CD25, FOXP3 type. These are critical for both inducing and maintaining tolerance. And they distribute systemically so that they can suppress responses in extrahepatic sites. You also need some level of deletion of effector T cells through activation-induced cell death. And then there are certain immune suppressive cytokines that are needed for the Treg induction and also to suppress cellular immune responses. It also turns out that the more transgene expression you have, the more Treg induction you have, and so the more you enforce the tolerance. In fact, if you don't express enough, you may not be able to induce tolerance. Now, this approach is now useful not also for a variety of diseases, such as I told you about hemophilia, but also other genetic diseases such as the lysosomal storage disorders. And actually, Dwight Coburl, I want to refer you to his talk. He will go into more detail of how hepatic gene transfer can be utilized to suppress responses in therapy for lysosomal storage disorders, so you can do secondary gene transfer or enzyme replacement therapy in those patients. And Brad Hoffman's lab has shown, and they have another talk about this coming up on Saturday in the immunology session, that this same approach can be useful to treat autoimmune diseases as well. Now, if everything fails and you just have immune responses you can't get around, however you design your vector, however you inject your vector, if nothing else works, then you, as I mentioned with the steroids, you might have to go back to immune suppression. I do want to make one comment about the steroids because I'm sure the question is going to come up anyway. In the hepatic gene transfer where these transaminases are observed and then CD8 cell responses occur, there are also examples of transaminases occurring without measurable CD8 cell response. So the liver the toxicity in the tissue, it doesn't have to be because of the CD8 cell response to capsid. There could be other reasons, although that's clearly one of the reasons. And in those cases where you do get the CD8 cell response, having that sort of immune suppression in place is very helpful. But it's not that it'll, that you can say any kind of any type of toxicity is necessarily the result of that. And conversely, you might find patients that show activated CD8 cells against capsid and don't show the destruction of the hepatocytes, because just because the CD8 cell is activated doesn't necessarily mean it's going to do its job. And just because the T cell is activated doesn't mean that it's a CD8 cell unless you do further phenotyping. So it depends on your assay whether you can even tell that that's a cytotoxic T cell or not. So those are caveats you have to keep in mind when you're designing or in trying to interpret these data. But nonetheless, it's been very helpful in many examples. And then there's a number of protocols, I don't want to go through them all, that show the value of immune suppression against transgene and capsid. I just want to point out there's been much emphasis lately on rapamycin. Rapamycin is quite useful because it helps you shift a response from an effector response to a Treg response. And it has been shown to be able to, if rapamycin is given to the transgene product ahead of time, you can pre-tolerize. It has synergistic effect with other drugs been shown. It can be packaged into nanoparticles. That's been successful in blocking, even neutralizing antibody formation against AAV. And it's also been shown to be able to prevent and reverse antibody responses when it's combined with other drugs such as anti-CD20. And with that, I want to conclude and thank the audience. I hope you find it interesting.
And we have time for a few questions. The microphone for anybody who would like to ask a question is here up front. And Roland, if you don't mind, I'll actually start with one. So we're hearing a lot about capsid engineering from a number of groups, and I wonder if there's accumulating experience about whether that approach has been useful for modulating immunogenicity in two ways. First, is it possible to avoid pre-existing responses with an engineered capsid, even to the same tissue? And the second would be, are there approaches that people are using to engineer capsids to entirely evade neutralizing antibody responses using depletion of, for example, capsids with immune sera?
Well, the first one is for sure that you can engineer the capsids such that maybe you won't be able to give it to every patient, but that engineer the capsid such a way that instead of, say, 40% of patients have neutralizing antibody, now it's only 10%. So I think that's realistic, and then hopefully we'll ultimately have a number of different capsids available so that there are choices, and then one patient can get that vector versus the other, depending on their pre-existing neutralizing antibody profile. The second part I think is very hard. The closest I have seen is probably this nanoparticle approach. How durable that will be and how that would work out for different vector doses. I think where all of that becomes very challenging is when you have very high doses you're trying to give systemically. But certainly once you've given the vector, you can't re-administer unless you go in through some, it looks like, the immune suppression ones look a little more promising right now than any of the engineering for that purpose, I would say.
Thank you. Question?
So you briefly touched about immune response, and recently Jim's lab showed that by giving high dose systemic AAV9 or AAV9 family capsid, they observed the acute kind innate-like response in the non-human primate. But Jerry's lab has done that in the human patients, in SMA patients, without seeing that. I was just curious, what's your thoughts on that? Is this something specific for non-human primate, or was it more?
Yeah, I think it's very hard for me to answer that question because, I don't think it's known yet in those primates what that innate, if that was an innate immune response for sure. It does look like it, but, and then against what? Against the capsid, the virus, something else in the prep. And then in patient studies so far what's been done, there were certainly some toxicities there, but they could be managed by steroids. So I think it's very hard for me to say because these studies seem to me somewhat preliminary and they have few animals, and I would like them reproduced also by somebody else. I think I would be purely speculating, trying to give an answer to that.
Quick, John?
Yeah, just a quick follow-up to that, John Gray. I appreciate those comments. I think they're very wise. Is it true, wouldn't an innate response be less variable between individuals? I mean, if only one of those animals out of 3 was giving that severe response?
That is a good question as well. I don't think you could say that, that you couldn't have variability in innate responses. In our inbred strains of mice, we don't have, but, non-human primates aren't inbred, so I wouldn't necessarily say that. But in terms of what drives the response, I think there was some interesting discussions in those papers that I didn't fully understand because if it's an innate response, it's not antigen specific, so it would be recognizing patterns. So, we can have a long discussion about that, but So how to interpret that based on the results given wasn't clear to me because you should be recognizing patterns, or there's something about the genome or the, so I think there were differences also from one study to another and not only between animals that it goes a little bit to your question. I mean, of course there can be variability in the severity of the response from one animal to another, but there should be certain commonalities that I didn't see between the studies that made me wonder what exactly is going on.
Thank you, Roland. Our next speaker,